welcome to another exciting episode of Teach Me in 10. Today we're joined by Dr. Mitzy Rios and by Georgina Miller, who are both scientists at bit.bio. They're both part of the team that developed the IO astrocytes that are the topic of today's video. And bit.bio is a synthetic biology company focused on generating human cells, advanced research and drug discovery. Today, Mitzy and Georgina will talk to us about how bit.bio's latest cell type, IO astrocytes, can help to better model complex CNS biology in vitro and ultimately improve drug discovery outcomes. If you'd like to learn more about bit.bio, you can click the link below in the description. Hi both! So before we start, maybe we can spend a little bit of time on bit.bio. Can you tell us a bit more about it?
Well, at bit.bio, we are focused on synthetic biology. We are a company that wants to provide human cells for research, drug discovery, and cell therapy. I believe what it makes outbreaking at work is that for a long time, the use of human cells in research was a privilege that was reserved just for a select few scientists. But we have revolutionized this by industrializing the generation of human cells through a new programming paradigm. This is relevant for therapies. For instance, the use of these cells can be used to repress other damaged cells, for example, organ systems, and eventually maybe we can replace entire organs. For drug development, the use of these human cells has the potential to significantly accelerate the process by providing the tools for systematic testing in human biology, addressing the bottleneck of clinical translation. I believe that right now there is a tremendous amount of momentum in our work, and we believe that a lot of people can access and use these cells. So it's an incredibly exciting time to be part of this transformation.
A key innovation from bit.bio is the development of OptiOx, which stands for Optimized Inducible Overexpression, which is a system that enables us to manufacture human cells with consistency and at scale. We're also currently building a clinical pipeline and providing a wide range of wild type cells and disease models for research and drug development. These are called IO cells, and they include nerve cells, immune cells, and also muscle cells. But we're really excited about a new product we've just launched, astrocytes, which now completes our toolkit of CNS cells.
Nice. So astrocytes are one of the cell types that bit.bio is working on, as you just mentioned. But could you tell us a bit more about astrocytes, where they're found in the body, and maybe how they work?
Yeah, absolutely. Astrocytes are fascinating cells. They're actually the most abundant and diverse glial cells within the CNS, and their name comes from the Greek words astron, which means star, and kytos, which means hollow vessel or cell, which makes sense when you look at them because they're star shaped. So they have these long branching processes which all come from a central cell body or soma. And what's really interesting is that their star shape actually correlates to their function. So they have these long processes which can connect then with hundreds of thousands of synapses within the brain, so provide neurotrophic support. But they're not just there to help neurons, they're also there for a wide range of functions, including glutamate homeostasis, so they prevent excitotoxicity, which can be really damaging to neurons.
Yeah, and as Georgie mentioned, their functions are wide. Another one we can mention is how they are key players in regulating inflammation and are involved in phagocytic clearance, meaning that they can help clean up the debris in the brain. Additionally, they play a crucial role in forming and maintaining the blood brain barrier, and they work along other cells such as pericytes and endothelial cells to protect the brain from harmful substances, and they also make sure that there's proper blood flow. I believe that we can summarize that say that astrocytes are quite diverse. They have different shapes, expression profiles, functions, and this all depends on where they are in the brain.
Got you. So you mentioned about astrocytes interacting with other cell types. Could you maybe elaborate further on how they're interacting with other cells?
Definitely, astrocytes are involved in a lot of different interactions and several networks, but there are four main ones that stand out. First, they interact with other astrocytes, so they create networks that help synchronize activity within the brain and regulate homeostasis. This is fundamental for proper cell signaling and supporting neural circuits. And talk about neurons, this is another interaction that is crucial. Astrocytes and neurons interactions are important. For instance, astrocytes release synaptogenic molecules that are essential for development and maturation of synapses. But also, once the brain is matured, these astrocytes continue to support neural activity by providing metabolic support to synapses, which is crucial for maintaining healthy brain function throughout adulthood.
For sure, but let's not forget about their role in myelin production and maintenance, which they achieve with their interactions with the oligodendrocytes. This relationship is super important for keeping the iron balance in check and providing the necessary metabolic support for myelin, which is the insulating layer around neurons. Another key interaction is with microglia, which are the brain's immune cells. So, astrocytes and microglia can actually work together to maintain homeostasis, clear debris, and also manage inflammatory responses. These interactions are often mediated by secreted molecules, something like growth factors, cytokines, or even neurotransmitters.
And last but not least, we should mention their interaction with endothelial cells and pericytes. These interactions are fundamental for the formation and integrity and maintenance of the blood-brain barrier, which is crucial for regulating blood flow in the brain and keeping the central nervous system environment stable.
Nice. So, sounds like these sort of cellular networks are very complex. What happens when things go wrong, and what are the associated diseases and implications?
It's a really important question because, as we've said, astrocytes have such an important role in supporting and maintaining the brain's environment. So, you can imagine when they don't function properly, it leads to a whole host of issues. Some of the most significant can be neurodegenerative diseases like Alzheimer's, Parkinson's, and amyotrophic lateral sclerosis, or ALS. And even though research is ongoing, we do know quite a lot about what happens when things go wrong. So, something specific to astrocytes is called reactive astrogliosis, which essentially is when astrocytes become activated by either an inflammatory response or an injury. When this happens, they start releasing pro inflammatory cytokines, which can cause a lot of damage. This messes with glutamate homeostasis and disrupts the blood brain barrier, which, as you can imagine, is really bad news for neurons.
And another area that's getting a lot of attention, especially in the context of Alzheimer's, is how astrocytes handle, or rather fail to handle, phagocytic clearance of toxic amyloid beta plaques. And normally, astrocytes help clear these toxic substances, but when they're not functioning properly, these plaques build up and contribute to disease progression. We can say that in a nutshell, astrocytes are brain caretakers. They keep everything running smoothly, so when they're not doing their job, whether it's due to an injury or neurodegenerative diseases, things can go down really fast and really bad. I think that's why astrocytes are implicated in such a wide range of diseases, and it's their role in maintaining brain's balance is absolutely crucial.
Hmm. Absolutely. So, given the complex systems these cells form, how can this be studied in the lab?
Oh, that's a great question. While we've learned a lot in the past about astrocytes, there are still so many unanswered questions, especially when it comes to the different subtypes, their specific functions, and how they are involved in various diseases. I believe that one of the biggest challenges has been the lack of physiologically relevant models. I mean, the traditional methods like direct differentiation are not only time consuming and expensive, but they often don't give us the most accurate human astrocytes to work with. Also, there's been a lot of work in relying on rodent astrocytes, but they are quite different for human cells, so they are not ideal.
Yeah, exactly. So to tackle this at bit.bio, we've been working on human-derived astrocytes. And these are a game changer because you can manipulate them in vitro and they can be cultured on their own, so in monocultures, but also along other human iPSC-derived cells. So it's really exciting to see how these astrocytes can help us understand so much about their roles, both in health but also in disease.
And I believe that it's not just about understanding their basic functions. We can create in vitro environments that simulate what happens during an injury or a disease by introducing inflammatory signals to the astrocytes. This can help us uncover new pathological mechanisms and potential pharmacological targets, which is really important for developing new therapies.
Yeah, and the possibilities don't stop there. Astrocytes are also proving to be super useful in drug discovery. So we can observe how they respond to new pharmaceutical agents, which could then maybe lead to breakthroughs in treatment. Plus, because astrocytes interact with so many other cell types, as we've just mentioned, we're able to actually mimic these interactions in the lab with co-cultures. So at BitBio, for example, we've been working on protocols to study the interaction between astrocytes and neurons. And this is really important for creating a more complex CNS model, but also it enables us to improve the activity of our neural networks in iPSC-derived neuron cultures as well.
Yeah, I believe that we are confident that our IO astrocytes are going to be an invaluable tool for researchers in biotechnology, industry, and academia who are exploring the astrocyte biology and looking for new therapeutic and regenerative interventions.
That sounds incredibly promising. Could you tell me a bit more about what makes BitBio's human iPSC derived astrocytes so unique, and how do they help scientists get a better understanding of complex CNS biology?
Absolutely. If I'm honest with you, in the past, working with astrocytes used to be such pain. There were a lot of hurdles, but with our astrocytes, we managed to address some of these biggest issues. I said there are three key advantages. First, since they're derived from human induced pluripotent stem cells, these astrocytes are much more physiologically relevant compared to rodent astrocytes, which have been the gold standard for so long but aren't the same as human astrocytes. They differ in size, morphology, expression profiles, and functions, which can make a huge difference in research outcomes.
Yeah, for sure. And another big advantage is our Optiox technology, which means that we can produce a really pure population of astrocytes. This is a massive deal because normally traditional methods, you have a purification step to get rid of infiltrating neuronal or other glial cells, which you then have to spend a long time and also a lot of money to purify them, only to then get a heterogeneous mix in the end, which can't really be used for experiments.
And let's not forget about consistency. With astrocytes, we've achieved a lot to lot consistency, which is a huge relief for scientists. You can do multiple experiments using different vials and still get reliable results, which is something that was hard to achieve with other methods, where batch variation was a big issue.
Yeah, so all in all, the human background, the purity, and the consistency of astrocytes offer an excellent model for studying the complexity of the CNS. Also, the faster protocol compared to traditional methods is just another massive advantage that the cells provide. It's a super exciting time to be working in this field.
Absolutely. So, how can scientists who are watching this video access astrocytes or find out a bit more information?
Well, all of our data, including the co-culture protocol, it's available in our website. In terms of accessibility, IO astrocytes are currently available under an early access program, offering researchers an opportunity to integrate these advanced cells into their work. Cells are in stock, ready to ship, and are supported with both mono and co-culture protocols. Further information about these cells is available in our website. However, our team is also on handy and happy to support any questions anytime.
Wonderful. Thank you ever so much for joining us.
Thank you.
Thanks so much for watching today's Teach Me in 10, and I hope I'll see you again very soon.